Hey everyone, so we are here today to do a very cool lab. You can actually do this lab at home. It is 100% safe. You should have all of the supplies around your house, hopefully. But we are going to be extracting DNA from strawberries. So before we get started, just a reminder about safety. If you have long hair, please pull your hair back out of your face. You don't want it falling into your experiment or getting your experiment on you. Please make sure you've got them closed-toed shoes. We got the Crocs on today. Any danglies, long necklaces you have, make sure they're tucked away. Again, you don't want it either getting into your experiment or getting on you. And that should, I think, cover all of the safety that we need to do today. If you have glasses, I suggest putting glasses on your face as like a safety measure, but really we're doing like kitchen type stuff, okay? So like I said, we are extracting DNA from strawberries. So for this lab, we are gonna need some equipment. You will need some strawberries. I have some fresh ones. You can use frozen. I don't think they put strawberries in a can, but you can use canned strawberries as well. They all work. The cool thing about strawberries is that they are what we call octoploid. So oct or octo means 8. Ploid is talking about the number of copies of chromosomes. So strawberries have 8 copies of every single chromosome, which means they have a lot of DNA inside of them. As a comparison, humans are diploid. We have 2 copies of every chromosome. We get one from mom, one from dad. And so diploid, 2 copies of each one. Strawberries have 8, they're octoploid. So for us, that just means they have a lot of DNA inside of them. So besides strawberries, you will need a plastic Ziploc bag. You will need some soapy, salty water. So this has some dish soap and some salt and some water mixed into it. You will need a cup. I'm using a glass cup. And you're supposed to have a funnel. I couldn't find my funnel, so I'm using a blender cup top. We have a couple of these around the house, so I took the top off and I'm gonna use it as a funnel, making it work. And you will need a coffee filter for your funnel. That's gonna be set up like that. I will come back to that. And then you will need some rubbing alcohol, and it should be in your freezer. So I have another bottle in the freezer ready to go. All right, so if you would like to follow along at home, that is the stuff you will need. If I'm being completely honest, I could have measured out the soap and salt that I put into this water. I did not. I just squirted some soap in. I sprinkled some salt in, did the little salt bay thing, and I mixed it up. It should work. I've done this with my 7-year-old before, and it came out perfect. So we should be good to go. All right, so like I said, step number 1 is we take our strawberries and we are going to put them into our Ziploc bag. You're gonna zip it up nice and tight, get all the air out of it. And what we are gonna do is just squish up these strawberries. You wanna get it as liquid as possible. The idea behind squishing up the strawberries right now is we are starting to break up the cells that are inside of the strawberries. And remember, DNA is stored in the nucleus of the cell. So we have to break down the cell membrane and the cells of the strawberries in order to start to access the DNA. So like I said, I am just mushing this up. I want it as liquid as possible. You do not want any chunks left in here. I used 3 strawberries. You can use like 2 to 3 strawberries. It should work. Like I said, this is a pretty simple lab to get results from, and it's pretty cool. So yes, are we all mushed up? No, not yet. I will say, and I've seen this happen many more times than you might expect, just be careful when you are mushing your strawberries. It's a plastic bag. It can pop open, it can rip. You do not want strawberry juice squirting all up on you, your kitchen, your clothes, whatever it is you're doing. All right, so here is my strawberry juice right here. Let's get a close-up of it. Ta-da! Okay, so from this point right here, again, if we're doing this a little bit more technically, we should have a graduated cylinder where we can measure out how much of our soapy, salty water that we're using. If we're talking science, this is a DNA extraction buffer. I know it sounds super fancy, but what I'm gonna do is I'm gonna try to keep the bubbles out of the bag, but I'm gonna pour a bunch of this liquid inside. That should be good. So you can see how much liquid I added. Like I said, your measurements don't have to be completely accurate for this experiment. That's kind of the nice part of it. The reason why we are adding soapy, salty water to our strawberry juice, this DNA extraction buffer. So DNA extraction, we're trying to extract the DNA from the strawberries. The soap and the salt will help to break down the cell organelles inside of the strawberry cells. And so remember, the DNA is located inside the cell organelle called the nucleus. And so we need to break down all the nuclei in our cells in order to get the DNA out of the strawberry cells. So again, I'm squishing this around. You want to do this for a couple minutes. Try to keep your bubbles to a minimum. So that's a pro tip right there from my years of experience of extracting DNA from strawberries. Keep your bubbles to a minimum. This just helps the filtration process go a little bit faster and a little bit more smoothly. All right, so I think we are good. We are mixed up. We are ready to go. We are very liquidy in here. So yes, looking delicious, right? So at this point in this bag, we have all of our cells, we have all of like our cell organelle gunk, and we have our DNA. So what I'm gonna be doing is I'm gonna be using the coffee filter and the funnel to basically try to filter the strawberry DNA away from the rest of the gunk that is in here. And that's what this filtration is going to do. So I'm gonna pour my strawberry juice gunk into my funnel. Oh, obviously we're gonna make a mess. I'm gonna pour it into my funnel right here. You don't wanna like overload it. Yep, that seems good. So you might have to do this in like a couple bouts, but basically this is a slow process. You can like set it and forget it type of thing. And you're just waiting for the strawberry juice to filter on through to the bottom of the cup. What we're collecting down here is where the DNA will be. So like I said, we're gonna let it sit for a while, let it drip. Please do not like poke around at it, stir it around. Do not try to make this process go faster. Be patient with it, let it sit. If we were doing this in a classroom together, we would usually have like a 90-minute block to do this, and we would spend the majority of that 90 minutes just letting this filter through. So that's what we're gonna do. You can see it dripping right here. I'm starting to collect some. I'm gonna turn the camera off because you don't need to watch this filtration process. It's like watching paint dry. But I will check back in once I have collected all of my strawberry DNA in the bottom of my cup. Hey, so we are back. I wanna give you guys a close-up of what's gonna happen in this cup. So what we did is we filtered all of the DNA from our strawberry solution sludge mixture, and the DNA should be in this cup right now, but we have to separate it from the rest of the liquid. So I did forget to mention earlier that you will need a popsicle stick or like a wooden skewer. Wood works better than metal. You can still get it done with metal, but wood is better if you have it. So if you have like a popsicle in your freezer, go have a snack, eat your popsicle, take the stick, rinse it off, and then bring it back to your lab. So what we are going to do is I'm gonna take the rubbing alcohol and I'm going to, it's called decantering, but what I'm gonna do is very gently and carefully, I'm going to pour some of this alcohol into the cup, and you want to pour it down the side so it doesn't like mix in with the strawberry juice necessarily. And I'm going to let it sit there for a second. And you should be able to start to see the DNA forming inside of the cup. You're also going to see two different layers. So you're going to see the alcohol layer and then our strawberry sludge layer in there. And I'm hoping that with this close-up, we'll actually be able to see it. I'm starting to see some of it appear. It does take a minute or two. I'm not gonna say what it looks like. I'm gonna see if you can see it. I don't even know if this camera angle gets it very well, but we're trying. So again, I'm gonna let it wait a minute. Maybe I'll do some video editing magic here to jump ahead. There we go. So it's like that white kind of filmy stuff that you're starting to see. That's going to be our DNA. Again, you want to be as gentle as possible with this process, but I'm going to go in and collect the DNA and pull it out so you can actually see what it looks like. Keep in mind, the more you're stirring the pot, so to speak, the more that you're stirring around in here and disrupting both fluids, the more you're going to break down the DNA into smaller and smaller pieces, and that means it'll be harder to collect it for yourself to then go look at. So what I'm gonna do is I'm going to gently go around in a circle to try to collect all of the DNA, and then I'm gonna drag it up the side of the cup. And we're gonna see how much DNA we collected. So let's do this. This is the fun part, right? Where we actually get our results. Ooh, I can see it moving. It's swirling. Oh, it's swirling. Come on. Oh, we're getting some. We got a little bit. Come here. I can see you. I know you're there. We're gonna get a little bit. There's a lot in here. It's just not collecting. Can I twirl you around the popsicle stick? Welcome to science, when it's supposed to work and it doesn't always work. I mean, it works, but there we go. Twirl you around like some cotton candy. There we go. There we go. Now we're collecting you. Oh, this hopefully should be a good one. All right, so the technique I'm using right now is just twirling the popsicle stick around and collecting all. Oh, come on, fell apart. Anyway, I got some of it. There we go. So there's your close-up of our DNA product. So all of that gunk right there is what the DNA actually looks like. I'll leave your observations up to you, but this is the part where you would fill in the results section for what the DNA actually looks like. So there's your DNA product. If we refocus, you should still be able to see there's like white stuff in the cup. There we go. So the white stuff that's over here where my pinky is, that's some more DNA. I just couldn't get it on the popsicle stick for whatever reason. Maybe we can like look into the cup together. Oh yeah, there it is. Okay, focus. Yes, I believe in you. So right here, this white stuff right here is more DNA. I just couldn't get it on the popsicle stick. So let's leave that there again so you can make your observations about what the DNA actually looks like. Sorry if I'm making anyone dizzy. All right, but that is your result. As far as cleanup goes, I threw the coffee filter, the strawberry stuff, I threw it all out. It's perfectly safe to throw out. This can go down your sink. You can obviously throw your popsicle stick out too. So like I said, very easy lab to do on your own. Oh, that was almost a good one. Almost a good one. See, this is why I need you guys here, because you're usually better at this than I am. But yeah, so that was the lab. Make sure you're recording what you observed in your results section, and then we will check back in for the conclusion section.